This video is about the neurobiological basis of autism spectrum disorder. Autism spectrum disorder, or ASD, is a cognitive condition that is a neurodevelopmental disorder that affects behaviour, learning, and socialization. The disorder involves a delay or alteration in neurological development. There is no one type of autism disorder, but a variety of subtypes exist to better diagnose the individual. These subtypes include pervasive developmental disorder, autistic disorder, childhood disintegrative disorder, and Asperger's syndrome. These are all on the spectrum of autism, with each having different types of characteristics, as autism is not a single disorder but a spectrum of closely related disorders with a shared core of symptoms. Neurology is the branch of biology that approaches the anatomy and functions of nerves in the nervous system. Hence, the neurobiological basis of autism spectrum disorder regards the brain and nervous system's efficiency within an individual that has an autism spectrum disorder. Identifying autism spectrum disorders in people can be difficult, as each person displays different symptoms and severities. However, there are some core characteristics of autism, such as repetitive use of words or behaviours, strong reactions to noises or touch. They may also avoid eye contact, fascinate over objects, and have delayed speech. Did you know males are 4 times more likely than females to have an autism spectrum disorder? There is no determined reason or clear cause to why some individuals obtain an autism spectrum disorder and others don't. However, it has been recognised to be associated with genetic and environmental factors. Autism spectrum disorder isn't as rare as you think, with 1 in 70 people in Australia having an autism spectrum disorder. There was also a 25% growth in prevalence in autism spectrum disorders between 2015 to 2018. The diagnosed cases went from 164,000 to over 205,000 cases in Australia. There are a number of regions of the brain that are involved in the cognitive alterations in individuals with autism spectrum disorders. Specific brain systems process information regarding social behaviour and reactions. Systems and corresponding anatomical regions that have been associated with neurosocial behaviour and seen to be affected in individuals with ASD include biological motion perception, which is linked to the superior temporal sulcus, face perception, which is linked to the fusiform gyrus, action perception symptom, which is linked to the inferior frontal gyrus and inferior parietal lobe. Perception of emotional states and emotional experiences linked to the amygdala and limbic system. Recent neuroimaging research has identified altered activity and disruption in these areas of the brain. There is also an overdevelopment of the brain growth in cerebellar brain areas. Neurotransmitters are chemical substances that are released by nerve impulses and affect the transfer of the impulse to another nerve fibre. There are specific neurotransmitters involved in human movement and the nervous system. It has been determined that some neurotransmitters in individuals with autism spectrum disorder are abnormal. Low levels of GABA have been observed in the left frontal lobe and cerebellum, which negatively influence development of local circuits that impact processing and cognitive behaviour. High levels of glutamate in blood plasma and serum have been reported in those with autism spectrum disorders, which is related to epileptic seizures. Abnormally high levels of dopamine have been found in the orbitofrontal cortex in individuals with high-functioning autism. All 3 types of neurotransmitters have been related to disruption of executive functioning, motor activity, social behaviour, and attentional skills. Neurological imaging has been a very crucial element of recent research and studies. Most evidence for atypical interconnectivity in ASD has relied on functional magnetic resonance imaging, or fMRI. Research has shown atypical connectivity in all individuals with ASD, whether that be underconnectivity, overconnectivity, and unusual patterns of connectivity. Other imaging techniques have been used to find neurological markers of autism. These include electroencephalography, which record electrical signals of the brain, and diffusion tensor imaging, which is a magnetic resonance imaging technique. Using a combination of these techniques, you are able to better understand why, where, and how autism spectrum disorder occurs. There are no current treatments for individuals with autism spectrum disorders. However, there is options to treat core symptoms, but are limited to psychosocial therapies, and some medications. Psychosocial therapies include applied behavior analysis and pivotal response treatment. These target communication, behavior, and social skills. The medications that are used help with anxiety, hyperactivity, inattention, and emotions. Most commonly used medications are risperidone and aripiprazole. However, there is no cure or treatment to eliminate an autism spectrum disorder. Research associated to understanding the neurobiological basis of autism spectrum disorder has increased drastically over recent years. This research has created a better understanding about neurobiology, genetics, early detections, and interventions that affect ASD. However, with these discoveries, an increase in prevalence has been identified and highlights the urgent need for further research. There is not one type of autism spectrum disorder, but many.